Welcome back to what is on the MCAT, Biology and Biochemistry Foundational Concept 2. Highly organized assemblies of molecules, cells, and organs interact to carry out the functions of living organisms. Cells are the basic unit structure in all living things. Mechanisms of cell division provide not only for the growth and maintenance of organisms, but also for the continuation of the species through asexual and sexual reproduction. The unique microenvironment to which a cell is exposed during development and division determines the fate of the cell by impacting gene expression and ultimately the cell's collection and distribution of macromolecules and its arrangement of subcellular organelles. In multicellular organisms, this process is necessary to maintaining life. The processes necessary to maintain life are executed by groups of cells organized into specialized structures within specialized functions, both of which result from the unique properties of the cell's component molecules. Content categories. Category 2A focuses on the assemblies of molecules, cells, and groups of cells within single cellular and multicellular organisms that function to execute the processes necessary to maintain life. Category 2B focuses on the structure, growth, physiology, and genetics of prokaryotes and the structure and life cycles of viruses. Category 2C focuses on the processes of cell and nuclear division and the mechanisms governing cell differentiation and specialization. With these building blocks, medical students will be able to learn how cells grow and integrate to form tissues and organs that carry out essential biochemical and physiological functions. What's on the MCAT? Section 2A, assemblies of molecules, cells, and groups of cells within single cellular and multicellular organisms. The processes necessary to maintain life are executed by assemblies of molecules, cells, and groups of cells, all of which are organized into highly specific structures as determined by the unique properties of their component molecules. This process necessary to maintain life requires that cells create and maintain internal environments within the cytoplasm and within certain organelles that are different from their external environments. Cell membranes separate the internal environment of the cell from the external environment. The specialized structure of the membrane, as described in the fluid mosaic model, allows the cell to be selectively permeable and dynamic, with homeostasis maintained by the constant movement of molecules across the membranes through a combination of active and passive processes driven by several forces, including electrochemical gradients. Eukaryotic cells also maintain internal membranes that partition the cell into specialized regions. These internal membranes facilitate cellular processes by minimizing conflicting interactions and increasing surface area where chemical reactions can occur. Membrane-bound organelles localize different processes or enzymatic reactions in time and space through interactions between proteins bound to the membranes of adjacent cells or between membrane-bound proteins and elements of the extracellular matrix. Cells of multicellular organisms organize into tissues, organs, and organ systems. Certain membrane-associated proteins also play key roles in identifying tissues or recent events in the cell's history for purposes of recognition of self versus foreign molecules. The content in this category covers the composition, structure, and function of cell membranes, the structure and function of the membrane-bound organelles of eukaryotic cells, and the structure and function of the major cytoskeletal elements. It covers the energetics of and mechanisms by which molecules or groups of molecules move across cell membranes. It also covers how cell-cell junctions and the extracellular matrix interact to form tissues with specialized functions. Epithelial tissue and connective tissue are covered in this category. Category 2A subtopics. Plasma membrane, general function in cell containment, composition of membranes, lipid components, phospholipids, steroids, waxes, protein components, fluid mosaic model, membrane dynamics, solute transport across membranes, thermodynamic considerations, osmosis, colligative properties, osmotic pressure, passive transport, active transport, sodium-potassium pump, membrane channels, membrane potential, membrane receptors, exocytosis and endocytosis, intercellular junctions, gap junctions, tight junctions, desmosomes, membrane-bound organelles, and defining characteristics of eukaryotic cells. Defining characteristics of eukaryotic cells include membrane-bound nucleus, presence of organelles, mitotic division. Nucleus, compartmentalization, storage of genetic information. Nucleolus, location and function. Nuclear envelope, nuclear pores. Mitochondria, site of ATP production. Inner and outer membrane structure. Self-replication. Lysosomes, membrane-bound vesicles containing hydrolytic enzymes. Endoplasmic reticulum, rough and smooth components, rough endoplasmic reticulum, site of ribosomes, double membrane structure, role in membrane biosynthesis, role in biosynthesis of secreted proteins, Golgi apparatus, general structure and role in packaging and secretion, peroxisomes, organelles that collect peroxides, cytoskeleton, general function in cell support and movement, microfilaments, composition and role in cleavage and contractility, microtubules, composition and role in support and transport, intermediate filaments, role in support, composition and function of cilia and flagella, centrioles, microtubule organizing centers, tissues formed from eukaryotic cells, epithelial cells, connective tissue cells. Category 2B. The structure, growth, physiology, and genetics of prokaryotes and viruses. The highly organized assembly of molecules that is the cell represents the fundamental unit of structure, function, and organization in all living organisms. In the hierarchy of biological organization, the cell is the simplest collection of matter capable of carrying out the processes that distinguish living systems. As such, cells have the ability to undergo metabolism, maintain homeostasis, including ionic gradients, grow, move in response to their local environments, respond to stimuli, reproduce, and adapt to their own environment. In successive generations, life at cellular levels arises from structural order and its dynamic modulation. This happens in response to signals, thereby reflecting properties that result from individual and interactive features of molecular assemblies, their compartmentalization, and their interaction with the environmental signals at many spatial and temporal scales. The content in this category covers the classification, structure, growth, physiology, and genetics of prokaryotes and the characteristics that distinguish them from eukaryotes. Viruses are also covered here. Section 2B, subtopics: cell theory, history and development, impact on biology, classification and structure of prokaryotic cells, prokaryotic domains, archaea, bacteria, major classifications of bacteria by shape, bacilli, which are rod-shaped, spiruli, which are spiral-shaped, and cocci, which are spherical. Lack of nuclear membrane and mitotic apparatus, lack of typical eukaryotic organelles, presence of cell wall in bacteria, and flagellar propulsion mechanism. Growth and physiology of prokaryotic cells. Reproduction by fission, high degree of genetic adaptability and acquisition of antibiotic resistance, exponential growth, existence of anaerobic and aerobic variants, parasitic and symbiotic, chemotaxis, genetics of prokaryotic cells, existence of plasmids and extragenomic DNA, transformation, incorporation into bacterial genome of DNA fragments from external medium, conjugation, transposons, also present in eukaryotic cells, virus structure, general structural characteristics, nucleic acid and protein enveloped and non-enveloped, lack organelles and nucleus, structural aspects of typical bacteriophage, genomic content, RNA or DNA, size relative to bacteria and eukaryotic cells, viral life cycle, self-replicating biological units that must reproduce within specific host cells, generalized phage and animal virus life cycles, attached to host, penetration of cell membrane or cell wall, and entry of viral genetic material. Use the host synthetic mechanisms to replicate viral components, self-assembly and release of new viral particles, transduction, transfer of genetic material by viruses, retrovirus life cycle, integration into host DNA, reverse transcriptase, HIV, prions and viroids, subviral particles. Section 2C, processes of cell division, differentiation, and specialization. The ability of organisms to reproduce their own kind is the characteristic that best distinguishes living things. In sexually reproducing organisms, the continuity of life is based on the processes of cell division and meiosis. The process of cell division is an integral part of the cell cycle. The progress of eukaryotic cells through the cell cycle is regulated by a complex molecular control system. Malfunctions in this system can result in unabated cellular division and ultimately the development of cancer. In the embryonic development of multicellular organisms, a fertilized egg gives rise to cells that differentiate into many different types of cells, each with a different structure, corresponding function, and location within the organism. During development, spatial temporal gradients in the interactions interactions between gene expression and various stimuli results in the structural and functional divergence of cells into specialized structures, organs, and tissues. The interaction of stimuli and genes is also explained by the progression of stem cells to terminal cells. The content in this category covers the cell cycle, the causes, genetics, and basic properties of cancer, the processes of meiosis and gametogenesis, and the mechanisms governing cell specialization and differentiation. Category 2C, subtopics. Mitosis. Mitotic process, prophase, metaphase, anaphase, telophase, interphase. Mitotic structures, centrioles, asters, spindles, chromatids, centromeres, kinetochores, nuclear membrane breakdown and reorganization, mechanisms of chromosome movement, phase of cell cycle, G0, G1, S, G2, M, growth arrest, control of cell cycle, loss of cell cycle controls in cancer, biosignaling, oncogenes, apoptosis, reproductive system, gametogenesis by meiosis, ovum and sperm, differences in formation, differences in morphology, relative contribution to the next generation, reproductive sequence, fertilization, implantation, development, birth, embryogenesis, stages of early development, order and general features of each, fertilization, cleavage, blastula formation, gastrulation, first cell movements, formation of primary germ layers, endoderm, mesoderm, ectoderm, neurulation, major structures arising out of primary germ layers, neural crest, environment-gene interaction in development, mechanisms of development, cell specialization, determination, differentiation, tissue types, cell-cell communication in development, cell migration, pluripotency, stem cells, gene regulation in development, programmed cell death, existence of regenerative capacity in various species, senescence and aging. Those are the concepts on biology and biochemistry on the MCAT for Foundational Concept 2. Up next, we will do questions for this set of topics. See you in the next video. For personal help, visit mcatboss.com or www.mattinmedicine.com. See you there.